Hi, everyone. Thanks for coming to my webinar today. I really appreciate your attendance and interest in the delivery of genetics and genomics at our health system and at our hospital and in our region. My name is Sandra Brown. I am a genetic counselor. My undergraduate was at University of Oregon in molecular genetics and biochemistry, and my master's degree is in clinical genetics with a specialty in genetic counseling. I'm board certified and also licensed by the state of California. I have been at St. Joe's and Mission Hospital for 9 years, almost 10 years. As we've merged with Providence, I'm now the regional manager of clinical genetics and genomics for Orange County and the High Desert. So I just wanted to start with a reflection. I really like this painting by Gauguin. And if someone speaks French, I'm sure I can't say what the title is in French, but basically, where do we come from? Who are we and where are we going? I really like reading philosophers, and especially discussing biotechnology and genetics. And Edward Wilson on Human Nature in 1978 said, "We are drowning in information while starving for wisdom. The world henceforth will be run by synthesizers, people able to put together the right information at the right time, think critically about it, and make important choices wisely." And if he thought in 1978 we were drowning in information, I don't know what he would think about today, but even more so. So if you think about all of the, our communities, our patients, we know that there are individuals in our communities that have higher risk for cancer, but we really don't know how to pick them out. And then ultimately, I think one of the goals of genetics is to determine who has lower risk for cancer as well. But where we're really focused and having the most success is in determining patients who have the highest risks, and then now also patients who have moderate risk of developing cancer. So being able to determine who these patients are in populations is the role of the genetics program. There's many publications around the inadequate delivery of genetic services to patients who meet these services. For example, only 10% of women who meet criteria at mammography would actually have a genetics appointment. And we know that patients who report their family history, that they do not really receive genetic services. And so the importance of their family history and the importance of their the management of their risk is not really obtained. We know that for patients who are diagnosed with cancer, specifically in this study showing that women diagnosed with breast cancer, of those that met the NCCN guidelines for genetic testing criteria, ultimately only 15% of those had genetic testing completed. When you look at women diagnosed with ovarian cancer, now ovarian cancer is a special case because there's no other criteria that needs to be met. Ovarian cancer diagnosis on its own is a criteria for genetic testing. And so in this case, you would assume at 100% that would meet criteria for testing, but you can see here that only 10.5% had genetic testing completed. Also, we know that patients who have next generation sequencing or somatic analysis of their tumor are interested in knowing whether or not that tumor identifies evidence that they may also be carrying a germline mutation. So we often want to look at those somatic results to see if there is germline evidence. So for those reasons, in developing the genetics programs here in Orange County, we have several ways of managing and identifying patients. We have well screening programs. So our well screening programs are predominantly in mammography, and we've worked hard to move them into endoscopy, but don't really have that set up in an efficient manner yet. But we've made great advances in mammography. We also have tumor-based screening programs. So our genetics programs at St. Jude, at Mission, and at St. Joe's screen every anatomic pathology report every week to make sure that we're identifying patients at the time of their diagnosis if they would benefit from genetic services and genetic screening. We also screen all of our molecular pathology reports, looking to see, like I was showing earlier, looking to see if there is evidence of germline within the somatic result. And to do this, we've increased our ability to provide additional services to patients for walk-in clinics. So patients who meet criteria can just walk into the genetics program. Our care program is through mammography. So patients who are identified through our care program can arrive in genetics and walk in for genetic testing, which includes oversight by a genetic counselor. For our pathology screening program, our patients can walk in and have genetic testing, again, and oversight by the genetic counselor. There's a lot of genes that we test patients for. Right now, we typically utilize an 80 gene panel to make sure that we're getting the right coverage. And all of these genes have relationships to risk, and understanding risk management, risk reduction is our primary role. You can tell when you look at this graph that there's a differential among the different genes on their relationship to risk. So on the very top left-hand side of the graph, you can see very high-risk genes, TP53, P10, CDH1. When patients have mutations in these genes, they have very high risk for cancer and need really extensive management, including surgical removal of at-risk tissue. And then as you go down, you can see here in pink, you can see the ATM, PALB2, CHEK2. These are genes that really give more moderate levels of risk. And then as you continue to go down, you can see lower risk alleles that are more likely picked up in polygenic risk. So when we're running polygenic risk analysis for a patient, looking to see if we can identify variants within them that are more associated with lower levels of risk, but then when you potentially add them up together, may cause significant increases in risk. And then there's also the interpreting of the genetic test result, really understanding if you see a variant of unknown significance, which is the VUS right in the center, is that an adequate characterization of an identified mutation? So there are levels of classification going up to pathogenic and going down to benign. And different mutations, variants can move through this field of classification based on the evidence. So the evidence that we use is typically based on reported personal and family history and collecting that evidence. And now we're using more sophisticated evidence, looking at protein analysis, protein function, RNA stability, things like that, that help us to classify these variants. But when we find variants in a patient, we will typically look and see what kind of evidence is there, especially if we find variants of unknown significance, what kind of evidence is there that's pointing that variant one direction or another to either help engage the patient's personal and family history to increase the pool of evidence for a laboratory to potentially make an adjustment in the classification, or to advise the patient of when we think they need to come in next so that we can continue to monitor a variant. So in doing this, all of these genetic services are important that they're connected with all of the other patients' care. That way, we're able to really improve their risk assessment, really improve our utilization, our management of the patient, patient and physician satisfaction, patient compliance, and get the outcomes that we are intending to get by, providing risk assessment and genetic services to patients. And so our primary role is to deliver genetic and genomic excellence, but we also enhance fluency across the communities. So we have lectures, presentations, support groups, different kinds of community outreach so that we continue to drive fluency. It's important that we all, when you think about even now with our COVID pandemic, it's important that people understand the language of genetics, that mRNA isn't necessarily a completely foreign word anymore. And so that's part of our goal, is to increase our fluency, to improve the awareness of our screening programs, to support programmatic developments. Any of our site programs, especially in our cancer centers, breast program, colorectal program, pancreas program, neuro programs, all these programs benefit from programmatic collegial developments that include genetics services. We also help to advise the health system and hospital goals. So I'm going to go over some of that a little bit later, and increase access to participation in genetic, familial, and genomic research. So for patient identification, we know that each cell type contains all of our DNA, but also is functionally utilizing different genes for each cell. And so for that reason, we've been able to figure out which genes are important in driving cancer that may be ubiquitous across all cell types. And we're continuing to increase the characterization of each important area of the loss of function or potentially gain of function within a cell that makes it more likely to be developing cancer. And it also gives us ideas of targeted therapies. So for example, if a cell has gained the ability to resist programmed cell death, then we know that resisting programmed cell death is an important area of potentially having a targeted therapy. And in the future, looking at CRISPR-like technologies, if not CRISPR, but CRISPR-like technologies to enhance the cell's function to be able to potentially restore some of these areas of loss of function, or to potentially hamper areas of gains of function. This is just a photograph of DNA. And of course, as you line up the chromosomes, having 2 copies of each chromosome, 2 copies of each gene, and looking at the genes across the chromosomes that are associated with inherited cancer risks. These are genes that we commonly test patients for to see if they have any inherent risk. And then the role of these genes, the role that they play in cell division, DNA repair, and survival. A lot of times the way that we talk to patients about them is that these genes are like little superheroes that help your cells to behave normally. And when you think about if a cell is allowed to grow, then it's a positive aspect. The cell is given a signal to go ahead and grow. And these genes are allowing growth. They're saying, go ahead and grow. We're giving you that green light to grow. But then as you go around cell division, cell cycling, there's the other side where there may be too much damage in the DNA, and then attempts to repair DNA and attempts to regulate, potentially negatively regulate cell division by programmed cell death and preventing cell division. So we know that most cancers are not inherited, but about 5 to 10% are likely due to high-risk hereditary mutation passing through the family. And about 30% are likely to be familial, meaning that we don't identify a high-risk mutation in the family, but probably more associated with polygenic risk, those low-level population variants that may be contributing to risk that you would see travel through a family, but you're not going to identify a high-risk mutation. So when we think about moderate risk genes and polygenic risk, they can blur a little bit. And you can also have a patient that carries a moderate risk gene and polygenic risk, or a patient who carries a high risk gene and a moderate risk gene, or high risk gene and polygenic risk. So all of those things really have to be considered together. And we really don't know what percentage of cancer risks are in these kinds of moderate risk or polygenic risk or combined factors. So I just wanted to go over, the most well-known and understood genes that are associated with hereditary risk. These are the BRCA genes, and you can see that the breast cancer risk is up to 87%. The second primary breast cancer risk is up to 60%. Ovarian cancer risk is much higher than the general population risk. And then prostate cancer, pancreatic cancer, male breast, and melanoma. And you can see these going down to very small numbers, like the melanoma risk of less than 1%. But that is actually dependent on which gene. BRCA2 tends to be higher in melanoma risk, and also the family history. So the family history could modify that, that risk higher as well. And then also genes, these genes are associated with pancreatic risk. And pancreatic risk is another one that's more associated with BRCA2 and is also modified based on family history. And then other genes that we commonly test patients for that are associated with breast cancer that may not be as well known. P10 gene is relatively rare. Carrying a P53 gene mutation is also rare. However, ATM, CHEK2, those two genes up to 60% and up to 48%, those are gene mutations we really find commonly. And then these are other genes associated with ovarian cancer. It starts out with the BRCA genes. STK11 is pretty rare syndrome, but we do see a lot of RAD51s, and also the Lynch syndrome associated genes, MLH1, MSH2, EpCAM. We do see BRIP1 and RAD51 CND. So these are genes that we commonly are identifying patients as carriers of. Cancer risks associated with Lynch syndrome. So Lynch syndrome is the most common syndrome or most common inherited form of colorectal cancer, but it also is the most common inherited form of uterine cancer. And you can see based on the gene here by this bar graph, MLH1, MSH2, and EpCAM give the highest risk for colorectal cancer, whereas MSH6 gives the highest risk for uterine cancer. And then each one has some contributed risk for ovarian, gastric, small bowel, especially ureter. We see those cancers coming in and identify these mutations. Pancreas again, and some other cancers, including central nervous system cancers and sebaceous neoplasms can sometimes bring a patient to our attention. Other genes associated with colorectal cancer are often associated with polyposis. So you can see biallelic MUTYH, biallelic meaning carries two mutations. Carrying one MUTYH mutation does not create this level of risk for colon cancer because it's not associated with polyposis. It does, we think it has a mild increased risk of colon cancer. APC is a classic gene known to cause polyposis, but it also causes attenuated polyposis. So it can be hard. You could see a patient with thousands of polyps and realize that they have polyposis, but you could also see a patient with fewer, maybe 10, 20 polyps and not realize that they have attenuated polyposis syndrome. And then you see the other genes here that are associated with colon cancer. And I don't see CHEK2 here, but CHEK2, oh, it's up in the corner there. CHEK2 is also associated, and that's a gene we find commonly mutated in our population. And then looking at prostate cancer. So we are, by NCCN guidelines, identifying patients. We are identifying all men with aggressive prostate cancer. So aggressive histology, Gleason scores higher than 7, metastatic prostate cancer. All of these prostate cancer types meet criteria for genetic testing. And these are the genes that we typically find associated with that a diagnosis of prostate cancer that may potentially have significant family history, but also may not have family history. And again, you'll see CHEK2. CHEK2 is an important gene for us. And ATM, those are genes that are more moderate in their risk, but we are identifying them more commonly. We always update the different programs, the different site programs for the updates in NCCN guidelines. So this was updated recently. There was also a colorectal NCCN guideline updated recently. This one is now breast, ovarian, and pancreatic. So they added pancreas to this specific familial guideline. And you can see that the guidelines for determining whether or not a patient meets testing criteria are quite extensive. There are 10 pages of criteria. And that's because the criteria are getting broader and broader, but still have detail. Eventually, I think we'll just be testing everybody, but while we're growing to have more broad testing, we're also still trying to maintain these criteria, which used to be 1 page and now they're 10 pages. And then you can see here, if the testing criteria is met, refer to the patient to genetic counseling. And then as you go along this decision tree, on the bottom right, you can see the patient was either not tested, they had a variant of uncertain significance, or no pathogenic or likely pathogenic mutation found. And this is a complex area for us that we spend the most time with our patients, because these are the areas where creating empiric risk estimates and risk management recommendations are the most complex. For the patients on the top that have positive mutations, creating or who have positive mutations in their family, but their test results are negative, those are the easiest because we have guidelines in understanding when we identify a mutation. When we don't identify a mutation, but there's still significant personal and family history, that's when the risk assessment, risk management is more complicated. So now the guidelines for NCCN are really built out for all of these genes that we commonly test patients for in these panels. So you can see that there is a lot of specific risk management associated, and they each have, many of them have their own pages in the NCCN guidelines. So when you're looking at the breast, ovarian, and pancreas cancer familial risk, NCCN guidelines, you can see now all of these genes are included in a table that give us updated information on patient management, and also have links to specific pages if there are pages for these specific genes. So it's gotten quite extensive, and the updates happen twice a year. There is also a list of genes that when they're inherited biallelically, meaning that if your patient carries one of these mutations, they may have relatively low, moderate, or high risk for developing cancer. But if they are considering having children, then it's important that they realize that if their partner also carries a mutation in one of these genes, then their children are at risk for autosomal recessive disorders. And some of these are cancer disorders, and others like Fanconi anemia are unique diseases of their own, and like ataxia telangiectasia as well. Constitutional mismatch repair deficiency syndrome, that's the 5th one down, you can see is associated with the Lynch syndrome genes. So we've seen quite a few patients, children with constitutional mismatch repair deficiency with brain cancers, because they carry a Lynch syndrome mutation from both sides of the family. So we always counsel our patients that carry these mutations that it's possible for a mutation to be inherited from both sides of the family. And so that's something that we look out for, especially if we see unusual diagnosis in children, or if we know that our patients are considering having children themselves, that they might want to have their partner tested to see on the low risk, especially if their partner doesn't have any family history, the off chance that there could be biallelic risk to their offspring. Like I said, the colorectal NCCN guidelines for genetic risk were also recently updated. And again, it's basically a list of genes that are associated with colorectal cancer, and guidelines, recommendations, and risk levels for each of these genes. So if it's something that you're interested in, or you have a patient that carries a mutation in one of these genes, this is kind of a good place to start to get additional information. So I also wanted to talk about our CARE program in mammography, which was a regional initiative And we started screening all patients undergoing mammography by sending them a chatbot 5 days before their mammogram. So if a patient is undergoing mammogram and you've sent your patient for a mammogram at Mission, St. Joe's, or St. Jude, they will receive this chatbot either through their cell phone or as an email to obtain their personal and family history information. So you can see there was an article in the Cancer Institute newsletter about it, and you can see that's kind of what it looks like when the patient holds their phone, is that it identifies itself as coming from one of the hospital breast centers, but also as a care program, and starts to interact with the patient to ask them questions about their personal and family history. And this is after 4 months. So we launched, you can see at the top here, we launched at St. Jude and Fullerton November 2020, at St. Joe's in Orange in January 2021, at Mission Hospital and Mission Laguna in February and March of this year. So because these were stepped launches, it's not a complete picture, but it's basically the January through April statistics. And you can see that we had 14,484 total mammography appointments, and that 79% of those patients completed their assessment. Of those, approximately 30% is what we now see as an average. 30% will meet the NCCN criteria for considering genetic testing or having genetic testing offered to the patient. And around 10 or 11% is what we see of that population will meet the Tyrer-Cuzick lifetime risk of breast cancer high risk score of greater than 20%. So that's a significant, actually it's a significant number, significant volume of patients, but also important to realize that this is a way that we are identifying these patients even before their mammogram, so that when they arrive at our breast imaging centers, that we're able to advise the patient that they're meeting these criteria, and that it's connected to their record rather than somehow only being obtained, or having it only happen once, or only being obtained through a genetic counseling appointment. It's something that's being auto-populated by the responses to the chat. And then you can see at this point that 556 patients had ordered genetic testing. And at this point, we had identified 55 mutations. So we do have around a 10% mutation detection rate in this program. When we're in mammography, or when we go into the portal, this is how we see a patient. I just took out the PHI. So you can see here, it's by the hour of their scheduled appointment for mammography. And so it's easy to go in as the front desk or the mammotech or the radiologist to see the line item of this patient includes their NCCN guidelines. The green checkbox just they meant their NCCN guidelines. And then their Tyrer-Cuzick score to see if they were above 20% and met that criteria. This little blue box here, it's just the basic risk assessment that's being completed by the application. So it's like a clinical summary. And so this is a portion of a patient's clinical summary. You can see at the top of the clinical summary, the patient did meet NCCN guidelines for genetic testing and did not meet Tyrer-Cuzick criteria of 20%. But you can also see that the patient's Tyrer-Cuzick score was 18.4%, which is relatively close to 20%. And we know that density does play a role, not only in modifying their Tyrer-Cuzick score, but also in breast cancer risk. There was just a publication out, I think just this week or last week, that was continuing to confirm the relationship between breast density and breast cancer risk. You can also see here the details of what the patient reported. So this patient reported that her mother had breast cancer at 55, maternal grandfather had prostate cancer at 65, and paternal aunt had ovarian cancer at 50. Now, this meeting the NCCN guidelines in this patient's case is because of the paternal aunt's diagnosis of ovarian cancer. However, the mother with breast cancer at 55 and maternal grandfather with prostate at 65 is risk inducing, but just doesn't quite meet that criteria threshold. So that is a patient, even if they didn't have the paternal aunt with ovarian cancer, could consider genetic testing, but does not obviously meet any criteria to do so. And then at the bottom, you can see the table that parses out the breast density and gives you the unique, for that patient, including their density, their Tyrer-Cuzick score. So you could see that if you were wondering about this patient, 18.4% lifetime risk of breast cancer, that's a little bit high, but doesn't quite meet criteria, then look at their density. If they were almost entirely fatty replaced, then their TC score goes down to about 7%. You might feel, okay, that's pretty good. You know, that means that my patient really doesn't have as high of a risk as is at first apparent by the TC score. But what if your patient was extremely dense? Then they actually do meet criteria for additional screening. So you can see an extremely dense breast would increase their TC score to about 22.5%. So far, we've had excellent feedback from this program. I do feel that it is a way to really deliver leading edge precision medicine to a large population. Right now, we are predicting that we will have screened 55,000 patients just in Orange County utilizing this tool, and utilizing one genetic counselor at Mission, one genetic counselor at St. Joe's, and one genetic counselor at St. Jude, 1 day a week to run the program that screens 55,000 women. And so I think that's an excellent conversion of productivity for our department, and allows us to really make a larger impact on the community as a whole. And also to support physicians in understanding their patients, the meaningfulness of their patients' responses to their family history questionnaire. And you can see we've gotten some very good quotes. And I think when you think about who are the users, like the lead mammotech at Mission Hospital saying that it's easy and patients love it, that makes it really rewarding to be running such a program, that the people who are on the ground using it, sometimes when we're designing programs and we're launching programs, we don't always understand exactly what that person, that caregiver that's on the ground with that patient will experience, and being able to see that that those caregivers are finding it easy and that patients love it is, I think, one of the most important quotes that I have here. Of course, this takes a lot of coordination from managing the IT to our scheduling departments. Hopefully remembering to tell the patient that they will get a chatbot with their before their mammogram, so that patients don't wonder if it's some kind of weird, malware or something that's trying to invade their cell phone, but that it's actually us trying to, coordinate their care and determine their risks. All the way through the front desks, the radiologists, the techs, people delivering the MRIs for the high-risk identified patients, the referring physicians trying to get all this information, and the genetics department as well. But if each person on this team does that small lift, it really creates this huge impact. I just wanted to go over to our pathology screening programs. So we do pathology screening for Lynch syndrome. So we screen all colorectal and uterine cancer diagnoses for Lynch syndrome. We also screen them for evidence of non-Lynch syndrome associated inherited cancer risks. We screen every ovarian, pancreas, every high-risk prostate. Those are just all pan syndrome screening, because they're just auto referrals to genetics based on guidelines. But we also screen every other cancer, so that we can find those rare tumors or rare associations with inherited risk that may not be apparent unless we look more deeply at the patient's chart. And we also do molecular profiling as well. So just a simple example. So screening for Lynch syndrome through IHC staining of the proteins associated with the same named genes that are associated with Lynch syndrome. This patient has absent expression of MLH1 and PMS2 proteins, and that is evidence that their whatever this is, colorectal or endometrial cancer, is caused by Lynch syndrome, meaning that there was a mutation in the MLH1 gene, or that there was some kind of a somatic event like hypermethylation of the MLH1 promoter region, which would cause the same effect. So it takes a little bit of screening to move through the workflow to get the right evidence and determine whether or not the patient would benefit from genetic testing. We also just recently, like trying to deal with Epic, have figured out, we used to have a Meditech program where we ran pathology queries in Meditech, but now we've had to migrate that over to Epic. And so we do run a pathology screening program in Epic where we use keywords to collect every week all of the patients that are the newly diagnosed patients with cancer. We also support the identification of metastatic and primary as additional keywords. If the pathology department would like to use those for reflexive testing to somatic molecular analyses. One of the things I'm working on with the health system that isn't ready yet is a much more sophisticated screening program. Right now, we're just adding the ability to add more functional keywords that would allow us to exclude things like negative for cancer. So right now we use the CAS list of, right now we have the CAS list, plus we have specific types of rare findings that are specific for us to identify patients who have high risk for inherited cancer or to be carriers of a germline mutation. And we need to have additional natural language tools that to help us to say, if the pathology report says no evidence of cancer, we don't want to pull that because we don't want to have to review it. So right now we are pulling those, and we want to be able to have a more functional pathology screening program that allows us to do more with this tool. So hopefully this tool is probably about another 6 months out before it's something that we can utilize and share with, potentially other teams that would benefit from having a tool like this. So one of the things that we do is we provide a molecular profile screening program. So some of the outside laboratories will pre-screen cases for us and send us lists when there is a somatic mutation identified that is also suspicious for a germline mutation. And then we screen these every week as well. And so you could wonder, how often when you were looking at a tumor and you found the somatic evidence of mutation, how often would that somatic finding actually be a germline finding? And you can see here that in some genes, it's quite common. So in the BRCA1 and BRCA2 genes, more than 75% of the time, if there's a mutation in the tumor, it's likely that it's actually germline in origin. And also the Lynch syndrome genes. Oops, sorry. And then as well as PALB2 and some of the other genes. So we have an idea of our suspicion based on these kinds of frequencies. So a gene like P53, we would rarely think that it was a germline finding. It's much more likely that it's a somatic finding. So we start to look at other evidence. For example, what's the patient's age? What kind of cancer do they have? Is it pathognomonic with one of the genes that are in the tumor? Is it, what about their family history? Is their family history also significant for some kind of inherited pattern? When we look at the molecular cancer genomics, I might not spend a lot of time on this unless there's a lot of interest, but it's one of my favorite areas. If you look at the relationship between the genes, the proteins that they build, and the way that they work in and out of our cells, nucleus, cytoplasm, transmembrane proteins, if you look at the relationship that they have with each other, and then you look at the mutations within these genes, it's just really fascinating to see how different DNA damages leads to cancer risk or cancer progression, but also provides clues for targeted therapies. So again, all my superheroes protecting our cells. And if you look at the BRCA pathway, it's kind of over here. And the BRCA pathway is really known for homologous repair. So homologous repair has to do with double-stranded breaks. So if your DNA is broken in both sides, so both strands are broken, it leaves a gap. And that gap is unwinding. It doesn't just sit there broken. It really starts to unwind, and the gap gets bigger and bigger. So homologous repair utilizes the BRCA1 and 2 genes and other genes that are in that pathway to really stop that unraveling and then bring the homologous chromosome. So the other, remember we looked at every chromosome, there's always two. So going to that other homologous chromosome and saying, you know, to that homologous chromosome in that area, there's a copy of that same gene. And so pulling it up, like parking it right next to that broken double-stranded break, and then using it to copy over the missing pieces so that there's no gaps. So when you have a BRCA mutation, you typically don't want to do end joining. End joining just means, okay, there's double-stranded break, you just glue it back together, and who cares if you create a big gap, which is, that big gap is known as loss of heterozygosity, and a lot of, like you might see that in a molecular tumor assay, that there's a loss of heterozygosity. That's what's caused by it, is that gap just being glued back together without fixing the missing pieces. So now you have nothing preventing end joining. So end joining starts to take place. So you're going to start to increase loss of heterozygosity, but you've also lost homologous repair. So as you lose homologous repair, what happens is that the loss of that's called the homologous recombination deficiency, or HRD. And as you have the loss of homologous repair, then you overload the system of end joining. And as you overload the system of end joining, you continually increase the numbers of these kinds of big gaps, loss of heterozygosity, that are in the somatic tumor, which are increasing the mutational burden of that tumor. So then ultimately, if you have a PARP inhibitor, when you have single-stranded breaks, the PARP inhibitor as a targeted therapy will increase the number of double-stranded breaks. And if you increase the number of double-stranded breaks in a system that's already overburdened by the inability to properly repair double-stranded breaks, then you basically overload the system. And when you overload the system, you cause the synthetic lethality for this cell. And that's why PARP inhibitors work so well, because they really utilize the function of the gene and the loss of function of the gene as the target. And then if you look over here on this side, you'll see the Lynch syndrome associated proteins at work with mismatch repair. And here they are, all these Lynch syndrome genes. And what mismatch repair is that these nucleotides, here you can see the G should not be lined up with the A. So the guanine and adenine should not be lined up together. So it should be T to A, G to C. That's the proper alignment for nucleotide bonding for chromosomal stability. And then at the top here, you can see a lot of times in our genome, we have these repeating sequences called microsatellites. You can see, you know, it says CTT, CTT, CTT. That's a microsatellite. And these are notorious for stressing the system. So if they stress the mismatch repair system, they're more likely then to be impacted by the loss of mismatch repair. So when there's loss of mismatch repair, you just have more mistakes in the microsatellite. So that's what's known as, that's what's known as microsatellite instability. So it's not really looking at the gene, but it's looking at the microsatellites for evidence that mismatch repair is not going that well. And then that causes a tumor mutational burden, because you can imagine if we're not able to repair these mismatches, then all over the tumor genome, there will be all kinds of mismatches. And that increases the number of aberrant proteins that are produced by the cell, which increases the evidence of tumor mutational burden and makes the cancer more likely to respond to immunotherapy. And I probably just skipped this one. It's just looking at transmembrane proteins and how they, if they're decoupled from the external growth signals, then they just cause constitutive growth without those signals. One of the projects that I'm working on is a virtual molecular tumor board. That's a tool, an application that we're launching next month that will be broadly available at the health system in order to basically ingest all of these kinds of molecular information and provide a platform and a venue to have a virtual molecular tumor board case conference. And you can see a little bit of what this tool currently looks like. One of the things that I think is the most important, because it is the genetic part that I helped to design, is to show the molecular changes over time in the tumor. So being able to show multiple biopsies analyzed, or liquid biopsy analyzed, or a tumor microenvironment analyzed, and being able to show that over time, juxtaposed to the patient's radiology, the patient's response to chemotherapy, so that you can get a real-time response to therapy or a resistance to therapy picture, and then have that include the molecular evidence. So when we identify that patients meet the criteria for genetic testing, especially in our pathology screening programs, we want them to have ease of access. We don't want them to have to go through a lot of hurdles to schedule an appointment in between their chemotherapy, radiation therapy, not feeling well. We don't want them to have to go through all those hurdles. So we designed this walk-in clinic so that the patient gets the pre-test elements. They're consented, educated around genetic testing. It takes around 10 minutes. We collect their saliva, and then a genetic counselor oversees their results and calls out their results and advises the patient. So when we initiated this project, sorry, this is a little blurry, you can see that a lot of different patients with different cancer types were utilizing the walk-in clinic. This is just our first 6-month data. We launched this in 2019, the end of 2019. So this is, I think, the first actual 9 months data of 2020. So I still have to put together data for 2021 for this program, but it's very successful. We've identified, you would think that there would be a big pie piece where we would identify really common mutations, but you can see we're identifying very important mutations, highly meaningful, potentially targeted therapy mutations across a broad spectrum of different genes. I also wanted to go over some of our other health system-wide access, I guess, for population screening or population genomics or genetic services. And this is the new Providence Genome for Me population genomics study. It's really predominantly meant for patients who don't meet criteria for any genetic testing, because if they do meet criteria, we want them to actually have specialized genetic testing that meets the suspicion or meets the needs of their diagnosis or the evidence of their personal family history. But this population screening program is an excellent tool for patients who are interested in having genetic testing and would like to participate in a free study that gives them access to the genetic testing. So this program is just recently designed and launched. And it's just meant to improve healthcare, to identify patients we wouldn't normally identify that have genetic risk. And it's available to any patient. If you're interested in knowing more, if you're interested in participating as a site, if you're interested in referring a patient, just send me an email or something and I'll connect you with the program. There's also a Sharepoint site. If you go to Sharepoint Genome for Me study, or Sharepoint for Providence Genomics, you'll see the Genome for Me link there as well. Genome for Me is basically looking at the ACMG 59, which are the most meaningful, a list of the most meaningful genes to test patients for, as well as the NCCN 47, as well as looking at, so most of these genes are associated with cancer or cardiogenetic risk. And then they also have the pharmacogenomics panel as well. So these are the genes that are associated on the Genome for Me panel. We also have a cardiogenetics program. You may not know that 1 in 200 people, it's about the same for cancer risks, have inherited cardiovascular disease. One of the most common is familial hypercholesterolemia. So making sure to test patients with familial or that have risk for familial hypercholesterolemia is an important area of genome for me, and also of being able to understand how to manage a patient that has high cholesterol levels and whether or not it's caused by this underlying genetic susceptibility. The referrals for genetics for cardiovascular risk basically include significant personal or family history, just like cancer. But you might see ICD implementation at an early age, like under 50, having histories of cardiomyopathy, arrhythmia, sudden cardiac death, or having evidence of other histories associated with inherited cardiovascular syndromes. The complexity of cardiac genetics is basically where cancer genetics was about 10 years ago, that there's a lot of overlapping features and there's a lot of variants of unknown significance, and the interpretation of risk is much more complicated and really requires a dedicated cardiogenetics genetic counselor to support and work together with the cardiologist to make sure that the correct evidence is obtained to call the interpretation of a genetic finding, or if there is no finding, to provide a risk assessment based on the cardiac evidence, as well as a family history. We also have genetic research that we're very much engaged with. One of the studies that we have open is basically a liquid colonoscopy. So we're looking for evidence of polyps through a blood draw. Right now, this study is for patients who are already planning to undergo a standard colonoscopy, and we are obtaining their blood sample before their prep so that we can use AI technology with this startup company that is creating a liquid colonoscopy. And what we're finding is that it's highly sensitive. It's not necessarily looking for circulating tumor DNA, because you don't want to have cancer when you're doing a colonoscopy. You actually want to know whether or not you need polyp excision. And so what the circulating evidence here is for looking at predominantly immunological signals that the patient's colon is developing a polyp. One of the other studies that we have upcoming is called Grail, or the Gallery trial. We'll have that launched. It should be launched by about October 1st. So we're just preparing to offer patients. In Orange County, we have such a highly developed genetics program that we're being offered the high-risk arm of the study. So you can see the problem here is that unscreened cancers, meaning cancers that we don't, or can't screen for well, represent 71% of cancer deaths. And that what we are identifying here is a test that, again, not so much using circulating tumor DNA, but in this case, we are looking to see if there's early evidence of cancer. Patients to participate in the study will not be suspicious for having cancer. They will just have high risk. So there will be mutation carriers, for example, that will have high risk, but they don't have suspicion for cancer. But we are looking for ctDNA because in this case, we are looking for evidence of cancer, but it's going to be very early stage. And we not only want to know where the cancer, or that they have cancer, we also need to know exactly where it is. So this test is, again, it's like a liquid screening. It can identify over 50 different types of cancer, where the cancer is located, as well as that the cancer exists. And the study includes the follow-up screening, so imaging screening to identify a diagnosis of cancer. So this is just a signal, so just a screening signal that the patient has evidence of hopefully an early stage cancer. If they do have cancer, that's the point of it. And also tells us where it is so that we can go look for it and find it. And all of this would be covered under the study. And again, the arm of the study that we have from the health system is the high risk arm. And this is just some more information. The false positive rate for this test is about 0.7%. So we do have a lot in place to follow patients and to provide the right workflow for the signal that this liquid screening is giving the patient. Future directions in genetics are obviously just more and more growth in precision and personalized medicine, including how do you add multimodal frequent genetic and genomic evidence. So how do you add multimodal and also multiple levels of different genetic evidence? Having partnerships that utilize AI learning tools so that we can make sense of very complex data and improve predicted responses to targeted therapies, especially when there's complex genomic evidence. Utilize things like CRISPR or mRNA or similar tumor editing to drive targeted therapies, and to use targeted therapies to reinstate a loss of function to help treat cancers, as well as clonal monitoring through liquid biopsy. So if a patient is completed with their treatment, using liquid biopsy to monitor the evidence that they may be developing a recurrence, or maybe the recurrence or the continued evolution of their cancer is adopting different clonal success. So something different where a targeted therapy would no longer work, or maybe a new targeted therapy could be identified. As well as risk management through liquid screening, as I just went over, as well as the growth of other types of the delivery of personalized medicine in the areas of cardiology and neurology. And that's all my slides.